Okay, let me go over this stuff. Some things that from the surveys, and I'll put them up. Still recording, yeah. Okay, some problems people have been having are basically that each class has a different format, so you can't remember where to find material from class to class, so send out the material. So I've been doing this with like the study guides, but I'll start sending stuff out. Remember, the lectures are on YouTube. I can't add my lectures to the eCampus. I have to search for them. So when I go upstairs and load the file, and the file is rendered, and then it's automatically put on YouTube. Sometimes it takes a day or so before I can find my own lectures. I can't copy and paste them, so I'm not going to put them on eCampus anymore. They'll be on YouTube. Quizzes open longer. I don't have a real problem with that. And instead of reviewing them in class, I'll review them online. And that'll open up a bit more time for the quizzes, and exams probably be open for more than one day. And a few people say they're having problems with using Hangouts, but I've not, I'm glad I did the poll because no one said anything. If you're all online, you can always get in touch with me, and we can go over it. One of the reasons why I use Google Hangouts is that there's so much information about how to use Google Hangouts, much more than any of the other systems. So if you're still having a problem, you can either contact me and we can try it out to be sure you're using. I've been doing Hangouts with groups now for a while. You can also send questions in to me by email, and then I'll post answers. The final will not, I had a question, when will the final be? The final is usually the last Tuesday of final exams at 8 in the morning. So I'll probably have it due that day, or by the end of that day, because I think that's right before finals are done with. And so you'll have a window for taking the final, and you'll have a window for taking the quizzes. Quizzes will be open. Final will be. I guess I didn't finish that. I'm not sure of the exact date of the final. I want to say it's always Tuesday at 8 a.m. So I'm going to disconnect for so what I'll do, this is another thing. So what I'll do for the exam is the day of the exam will be, the class before the exam will be a review. So either in class or somewhere, and then I'll set up more online times, okay, if you're taking the course online. Remember though, if you're taking it online, remember I've sent this out several times, you need to read the start of every lecture, because that's where I'm sort of covering this kind of a take-home. There are a few other things, and I'll put them together. Nothing. The biggest problem seems to be that people are having a problem with every class having a different format, and so finding the lectures or finding the handouts. Oh, another one was, since someone noticed that I do everything inside of Google Docs, is just to make that folder where I have the lectures open, and I can do that. I can send out and make that public, and you can look at them. And I didn't put that. Any other questions? Anything? Yes, sir.
Is exam 3 the light reaction?
Exam 3 is photosynthesis and metabolism. Exam 4 will start with growth and development. And I'll send that out specifically in an email. So exam 3 traditionally has the lowest average. And I've tried it in the past where I'll split photosynthesis and metabolism, and that tends to make the two exams then have lower averages. Unlike me, you guys just don't like to memorize pathways and reactions. I don't understand why, but and you'll have a bit of time. Oh, someone said about passing out the practice exams. I consciously don't do that until close to the exam time, because based on experience, and not just me, but when people have actually done the experiments, if you pass out a lot of old exams and you don't use the old exams to write the new exams, and remember, I don't use the old exams, but there's just so many ways you can ask things, so 50% of the exam will be the same maybe. Anyway, if you have like an old exam bank, and where you do it with and without, or where you do it where you give the old exam closer to the exam, people score higher on the exam, because you study the old exams and you forget, and you don't study the material, you study the exams. So I'll send those out. What I do is I go through a couple of old exams and I pull some stuff out and then send it out to you. Okay? Next question. Other questions?
So we'll have a quiz this Friday?
It'll be open starting this Friday. And, we do have classes Friday. Easter's been canceled. You ever heard my really bad joke? Yeah, they found the body. That's it. Maybe I don't get struck by lightning over that. But anyway, any other questions? Okay. So now we're gonna cover, for the next 2 or 3 lectures, we're going to cover hormones. And we're going to start today with what hormones are, and what plant hormones are have changed drastically over the last 20 years or so, or longer than that. And we now have much newer definitions for what a plant hormone is versus an animal hormone, but of course we're going to start talking about what hormones are. And today we're going to talk about auxins, probably finish up auxins today. Next time I think we're talking about ethylene if we get through auxins today. Auxins are probably the most ubiquitous of hormones that are around. If there can be one hormone that's more ubiquitous than other hormones, it's the one that's been studied probably the most and the earliest. If we start thinking about what hormones are, and of course all of our terminology comes from animal systems because we knew about animal hormones. We may have actually known about plant hormones before animal hormones, but of course all the terminology comes from medical work. And it's basically, how can you get differentiation in this multicellular organism? Remember last time we talked about seed development, in that one of the first stages of the actual embryo growth is called the globular stage, and that's just a round ball of cells, and they keep dividing and dividing, and if there were no hormones, then the organism would stay as just a globular ball of cells. But as these cells grow, and then there's some differentiation that starts, then you start getting gradations of hormones. And the gradation of hormones then allow cells to differentiate. Remember we talked about differentiation, where you're going from this parenchyma cell, this anything cell, to where we can produce a phloem element or a xylem element, or a cell is going to fate to become part of the leaf. No. And so the way most of these things are done are hormones. And if you think about it, any given place in the plant, any given cell in the plant can have a slightly different hormone environment than the cell above it or below it. There's a lot of different hormones. Some of them they're produced in the tips, they're produced in the growing tips, or they might be produced in the root. So there can be there's this gradation of all these hormones in the plant. They were initially discovered in animals. I think they were actually worked on in plants first, but before they were actually discovered. And in plants, one of the first researchers to actually do experiments was Darwin, and we're actually going to talk about his experiment today. And here are the definition of plant hormones when I took plant physiology, which was they're naturally occurring, of course. They are synthesized in a very specific place, and the site of action is always remote from the site of synthesis, and that turns out not to be quite true anymore. One thing that is true is that they are active in extremely low concentrations. These have slightly changed over time. Hormone synthesis can occur in different kinds of cells in plants. So if we go back to 1970s, we had a hard time measuring hormones. We didn't have antibodies, or we didn't have the kind of equipment we have now. So we would, of course, find auxins in the growing tips, because that's basically such a high concentration there, we could find it. And then the next, we found that auxins sometimes are synthesized when there's damage to the plant. It was easier to study animal hormones. And they can also sometimes act in the same cells where they're produced. And something that you're going to see, and something that's really important because we use hormones all the time, is that there's not always this concentration and degree of bioactivity. And what we'll see, and what you're going to see, and what we see outside, is a little bit of a hormone may cause one reaction. A lot of a hormone may cause something totally different. A little bit of auxin may cause something may promote growth. A lot of auxin may cause the plant to defoliate. We don't usually see that in animal systems. We don't have this sort of two bifunctional responses. You add a hormone, you add too much hormone, you may cause some problems, but you're not going to get a different result. So the first one we're going to talk about are auxins. What are some common auxins? IBA. What else? NAA. What's the most common auxin? Well, no. What's the most common one used? Anybody know? Something called 2,4-D. Probably is most widely used. Auxins, we always cover auxins first because it has a lot to do with a lot of different things. And it can be synthesized in multiple locations. Usually we think of it in apical meristems, and to get ahead of myself here, that auxins, one of the easiest things to do an experiment with is apical dominance. And if you come through and you pinch the top of a plant, you kill the growing tip, take off the apical meristem, you're drastically lowering the concentration of auxin, and that causes all the lateral shoots to start growing. So you get a plant that's bushier. And what you're doing is you're destroying the source of auxins, and auxins keep the plant, keep the lateral buds from growing. We're going to cover that again. That's something that's easy to do. Here's the first experiment that was done with auxins. So you take an intact seedling, and everyone knows this, you shine a light only one side, so it doesn't have light from all directions. And what happens is then the shoot tip bends, okay? So the question then is it bending toward the light, or is it bending away from the dark? And what's going on here? So what Darwin could do, and what Darwin did, was he was able to cut the tips off, and these are oats. And you know, if you cut this off, you still have a little tiny little thing that sticks up that's part of the true leaf. So if you cut that top off and you shine light only to one side, nothing happens. The thing does not bend. Okay? Let's do another experiment. If we take an intact shoot and we cover up the top, put an opaque cap on the top, just maybe put a piece of, what we do in the lab is put a piece of aluminum foil over, just, put it on the top. You do get some bending, but not nearly as much, okay? Because I like to call it reagent grade darkness, and if you put a piece of aluminum foil even over an oat tip, it still gets a little bit of light, so a little light's going to sneak in under here. So what it seems to mean from just these experiments, or this experiment, series of experiments, is that there's something in the tip that's causing this thing to bend. And then later on, Boyce and Jensen did these experiments where they used a piece, or he used a piece of mica. Anybody know what mica is? Mica is that very, very, very thin mineral. You get a sheet of it and you can keep peeling the sheets apart. And so you could very easily stick a piece of mica on either side. So if the piece of mica was put on the side opposite the light, nothing happened. If the piece of mica was put on the right side, well, there was bending that occurred. Okay? In another series of experiments they did, you can take the tip off, and then you can put a piece of gelatin and put the tip back on. And guess what? It would bend then. So, and these were some of the first experiments, there seemed to be something diffusing from the tip that you could collect in the gelatin and then would go through the gelatin. Okay? And you could also do a similar set of experiments where you put the tip on asymmetrically, and you would get bending then. Okay? Take the tip off, put it on this side, you would get bending. And if I go back here, and you're going to see this in a little bit, we can actually take this gelatin, we can put the tip back on, wait a little bit, take this piece and take the tip off, and move the gelatin around like this, and it'll cause the bend. And so we know what's going on here. And that is, what is happening is that auxins, and this is an experiment that really shows you here, is that if we stop the movement of auxins down the dark side, we don't get the bending. So basically the take-home message is that what's happening is that when the light causes the asymmetrical distribution of auxins, which causes this side to elongate more than this side. So you get bending toward the light. So in the case of shoots, you have auxins inducing cell elongation, and that's what auxins do, and this goes back to the first week of class, or the second week of class, when we talked about acid-induced elongation of the cell walls. Auxins causing this bending. Later on we'll see that in the roots we get a little different response. Okay, the most common auxin is IAA, And there can be a wide range concentration of the hormone. Synthetic auxins include NAA, and other ones 2,4-D, 2,4,5-T, a commercial product here. And auxins are synthesized from an amino acid, and then they can also be synthesized by another pathway. Auxins, hormone synthesis here is, you don't have to worry about the 3 steps. Hormone synthesis tend to be, the last phases then, tend to be very short because we can see hormone responses incredibly quickly in plants. So they may be, and in the case we talk about ethylene especially, they may be ready to go, and they only need something to happen to get this immediate final stage of synthesis of the compound. So we get a quick response. So hormones can cause a quick response, you know. If we just sort of squint your eyes and look at them, they all look pretty much the same. Here is the very short synthesis from tryptophan. So you start with this amino acid, end up with IAA. 2,4-D is probably by far the cheapest hormone you can buy, period. 2,4-D is very, very inexpensive. Look at this, you look at this, look at this, you can see they're pretty much all the same structure. And so there are other formulations. This has become a real problem. This is, we talked about in the Midwest how they're now Roundup resistant. And actually I asked you about this, Roundup resistant weeds. So there's a move on, and they are developing 2,4-D resistant crop plants to switch from using Roundup to 2,4-D, or maybe a plant that has both Roundup resistance and 2,4-D resistance. And that's sort of scaring the people I work with a lot, because grapes are incredibly sensitive to 2,4-D. So since Monsanto also wants to produce the hormone, and 2,4-D of course is not, the patent ran out a long time ago, they've come up with a formulation of 2,4-D that's not nearly as, doesn't drift nearly as much as normal 2,4-D does. And I can't remember, in fact I don't know if they've actually somebody answered the question on the exam and I went and looked it up. I didn't know about it. It's a, can't remember now, but it's a modification of 2,4-D that really lowers the drift potential. And I can't even remember, I want to say it's a methyl, it's something, it's nothing major, but they can now patent that, and they can go back to sell you the plant material and also sell you the hormone to use, whether it's glyphosate or 2,4-D. Now, you've probably heard of 2,4-D, 2,4,5-T, and something called Agent Orange. And this was used a lot in Vietnam, and at high concentrations, 2,4-D, as you're going to see probably today, will actually cause a burst of ethylene production and cause the leaves to drop off plants. So you could defoliate, you know, hundreds and hundreds of square miles. People started getting sick, and I was in the next lab over when the lab which worked on birth defects and teratogens, and everyone in that lab worked on mice embryos, and what you do in that kind of work, it's pretty disgusting, terrible. We had our seminars at lunch, and you just couldn't eat lunch and watch these seminars. Rats, when the rats are pregnant, the fetuses are produced like beads on a string. So what you do is you feed the rats the carcinogen or a teratogen, and teratoma is a monster. So a teratogen is something that causes birth defects, monster maker. And then right before the the fetuses are at term, you, quote, sacrifice the rat, and you can pull out the fetuses and just basically look like pearls. And you can pop the first two out and stain the bones. You can take the second two and stain something else. And then you can do something with the nerves, I think always 8 or 10, something like that. And you can very easily look and see if they've got, the most common one is cleft palate. The second most common one, I can't even remember. Then you go to things like the brain, the skull is not formed. And then the first two, you drop into a solution that completely turns everything clear and stains the bones pink. So you can just pick them up and look and say, okay, there's some bone deformities here. And so it was in this lab next to the lab that I worked in where they actually found out it was actually dioxin that caused the problem, and that 2,4-D and 2,4,5-T were not causing the problems that the veterans were experiencing. But it was this dioxin, and dioxin is pretty bad stuff. And I saw there's a Arthur, I can't remember Arthur's name, he was at Yale, and he showed this seminar where they were loading up 2,4-D and 2,4,5-Ts. You know, as far as you could see, there were 55-gallon barrels of this stuff. You know, thousands of barrels of this stuff they were loading up and spraying daily. And they're right, you know, 2,4-D and 2,4,5-T are fine. I mean, I wouldn't want to drink them or anything, but they're pretty safe to use. But the dioxin was the byproduct they weren't cleaning up, and that's what caused all the problems. Now, if we look at where we find auxins in plants, in the relative amounts, we're going to find the most here. So this is where we first found it, of course. And then in the seed proper, we don't really find any. And then we start seeing a gradation, and we also get down to this growing tip. And in the root, we have auxins that are produced. Now, you can see that there's still some auxins produced below here. So this is a stylized plant. If we had a plant that had lateral buds, that each of those lateral buds would be a little bit of auxin. And if we take this part and cut this off so that we remove this auxin, then these level of auxins are just going to go increase because those lateral buds are going to start growing and producing auxin. And then that's also how you get the bushy plant. You cut the top here, and then these lateral dormant buds would start producing shoots, okay? The principal thing that auxins do is cell elongation. And I've repeatedly said most of the growth of the plant is due to not cell division, but after cell division, it's elongation of the cell and what the cell does then as it elongates. And we can increase it, and it's also saturable. In other words, you just can't keep adding auxin and auxin and auxin. You get to a point where extra auxin does not do anything, and we're doing this in a petri dish, and I'll show you that. So we can do some things that are pretty interesting with auxins. We can actually very easily, and we used to do this in the lab, we can do a bioassay. And before we had HPLCs and antibody tests, the easiest way of measuring auxin concentration was to take this is supposed to be a little gelatin, and you would take the plant material you were interested in and isolate as much as you could the auxin, and then put the auxin then in a piece of gelatin, and then put it on one side of the oak coleoptile, and then measure this angle, okay? So the more auxin you have, the greater the bend of the angle will be here. It's called a bioassay, and you should know that. And this is very accurate for auxins. Can't tell us what the auxin is, but it's fairly accurate. And an easier way even is to take and cut the internodes. So if we go back to our stylized plant, we cut this off, and so we take the area that's going to be the area of elongation, and we drop it in a petri dish with our different concentrations of auxin. And you can do this in an afternoon. So you can come to the plant phys lab, set up this experiment, come back in a couple of hours, and measure how much the stems have elongated. Okay? So the more auxin, the more elongation you'll get. And literally, you could do this in an afternoon, in a 4-hour lab. And this was the way that we measured auxins routinely until the newer equipment came in. Okay? One of the more interesting things that we find is the regeneration of vascular tissue. So auxins control the actual differentiation of tissue. And the one I think I always tell you about, and you can do this if you have a mint or anything that has a nice square stem where you can go in with a very sharp razor and cut a V of the vascular tissue out. You're going to see, like I said the last time, you're going to start seeing a callus form. You're going to start seeing undifferentiated tissue form. And then you're going to start seeing the vascular tissue start to differentiate. Phloem is lower concentrations, xylem at higher concentrations. And you can actually see this. So here's the wound. And here is the cut right here. And you see this vascular tissue, and then you actually see this tissue start to change appearance and actually reform into xylem and phloem. Well, why is it doing it here? The wounding causes a lot of things to happen, like ethylene is going to be produced, which we'll talk about later. Auxin is going to be produced. And so what happens in these cells, they have the right hormone concentration, not just of auxins, of maybe even GAs that are coming from someplace, or the different kinds of auxins. So they have that right environment to start differentiating the xylem and phloem and to regenerate that vascular connection. Okay? I've told you apical dominance. And auxins will actually suppress the cell mitosis and expansion. And we can remove it. And here you have a control plant. Here again, this is something you can easily do in a lab. Here you have a plant where you've pinched the top off, and now you get this lateral growth. And here you have a plant where you've pinched the top off, and it's hard to see that little blob there is lanolin, which is what we always use for some reason. And you've put auxin in that lanolin. So now you see you don't have these lateral buds being produced. So you cut the tip off, and then you replace the auxin source, in 2,4-D or IAA in lanolin, and you reestablish apical dominance. Okay. And there are also some chemicals we can use now, and we'll talk about those probably, where we can chemically reduce the apical dominance and cause the plants to bush out. Okay? Questions about that? And this is something you've seen. I told you the first of class that you'd keep seeing this over and over again, and that is that auxins cause a drop in the pH of the cell wall, which causes the cell wall bonds to break, and turgor then pushes the cellulose fibrils apart and increases the length of the cells. Okay, so plant cells elongate up and down before they do anything. And this is still termed the acid growth hypothesis. And it involves expansins, which we talked about. And somehow these expansins, in a method we still don't completely know, help control the length. And so what we can do is we can add IAA, and in this we're seeing pH drop, so we're seeing the pH go down with time, and we're seeing the elongation, the internode elongation, increasing. And if we go in here at this level, we add IAA, and we add a buffer to keep the pH from changing, which you can easily do, then of course we don't get elongation. Does that make sense? Or instead of adding IAA, I can lower the pH by adding a weak acid, and I'll get the stems to elongate, or the internodes to elongate. Okay? So here's the physiological response. We add IAA at this point. We see a lowering of the pH, and we see an increase of length. If I add IAA with a buffer to keep the pH from changing, I'm not going to get an increase in length. Okay? And here's what we think is happening, is that, and again you've seen this more than a couple times, is that the pH drop, the acidification of the cell wall area, is probably breaking some of these bonds. And these cross-linking bonds, remember, is what keeps the cellulose fibrils from continuously spreading apart. So if we start breaking these bonds or loosening these bonds, then remember, turgor is going to drive the cell to increase in size. It's going to push out on the cell, and these fibrils are going to elongate. Okay? And then the pH then goes back up, and then these bonds are reformed. So now we have a strong cell wall again that's not going to elongate. Here's another cartoon of what's happening. And this is, you can go look at this. This is from the NIH. Is you have the rigid wall, and you add auxin, and protons are exported, and it causes expansion. The hydrogen bonds are broken, and you have cell elongation. So here you have a system where you have the same pressure potential, but because of all the bonds, the cell wall is going to resist that pressure. With the auxins, you're actually weakening the bonds between the cellulose microfibrils, and so they're going to then respond to the turgor pressure by expanding out. And then it expands so much, and then the bonds are reformed. Okay? Otherwise, this would keep happening, and this cell would explode. Auxins move in more than one way. They can move from cell to cell, and we're also going to talk about transport. And the transport is bicipital. That means it's from the top to the bottom. They do move in the phloem, which we're going to talk about, I think, today. And they're transported from the top down. So bicipital means from the top down. Here's a neat picture where these segments have been turned upside down, and the roots are going to always grow at the bottom. So even if you turn these segments upside down, the roots are still going to grow at the bottom. Okay? So what's happening here is that the auxins are still being transported, in this case, from the top to the bottom, regardless of the orientation of the stem. And we can do this. We did this with one of my fellow students did this with kudzu. You set up an experiment where you have a block of radioactive IAA, and you have your stem segment, and then you have a receiver block. Let's look at the orientation. A is at the top and B is at the bottom. Okay? So we look over here, and we have B now at the top, so we turn this thing upside down. And so no matter what, even in the normal, you're going to get radioactive IAA, and even if it's turned down, you're going to get IAA. Okay? Oops, why doesn't that come up? I don't know what happened to that slide. Won't worry about that. Go to this one. Okay. This is basically the same thing. So what we think is happening is that we have multiple forms of IAA, and what you should look here at are these carrier proteins again. And you notice that they're only in this cartoon, they're only on the bottom side of the cell. So this is the tip up here, this is bicipital transport. So auxins can move in and out as either protonated or as the free acid. So they can actually be carried in, they can move in, but they can only be transported out of the cell by these carrier proteins that are on the bottom side of the cell. So if you turn this upside down so that this thing is completely inverted, then the auxins are still going to be transported from the top down. So they're going to be, if you turn this upside down, then auxins are going to be transported up because they're going still from the top down. Does that make sense? So you just have these proteins that are located on the bottom, and you can actually disrupt them and see what happens. Here is locating the auxin transport protein. And in this case here, this is a segment where this is the top, of course. The tip would be here, on down. And you can actually see that these proteins, and this is done by, you can add an antibody that lights up green. You can see they're on one end of the cells, and if you look real closely, you can see that they're actually on the bottom side of the cells. And then if you disrupt auxin transport, then these proteins, by disrupting these proteins, and these proteins are just sort of randomly arranged. So if you remove the proteins from the base of the cell, you're not going to get auxin transport. So this is sort of the simple diagram of what's going on here. Auxins can enter the cells in a variety of ways, but they're transported down out of the cell by proteins that are on the bottom side of the cell, if that makes sense. Okay, questions? So even if you turn this upside down, the auxins are still going to be transported on whatever side these are located. Okay? And I think I'll pick up phototropism next time since we're about out of time. Any questions? Okay. Yes, sir.
Auxins is just a byproduct from making Agent Orange. So it wasn't in the spray.
Yeah, it was in that 55-gallon barrel. They didn't purify it. Yeah.
So it was part of. It was like a byproduct in solution of Agent Orange.
Right.
Okay.
Agent Orange is 2,4-D, 2,4,5-T. I don't know why they call it Agent Orange. I bet it was painted. I bet it was orange colored.
Yeah.
And it was much later that they started looking at, when you synthesize 2,4-D, a byproduct is dioxin.
Yeah.
They didn't think anything about that.
Dioxin is what it was. So it
Yeah.
wasn't 2,4-D and it wasn't.
No, No.
So if you actually had those purified, like that chemical purified without the byproduct of dioxin, which is what we buy now at 4D at the store,
Yeah.
it's fine.
Yeah, yeah, I mean, yeah, there's a Wikipedia, well, anything Wikipedia, that, I'd link to that Wikipedia article about it.
Didn't Agent Orange work a whole lot better though than 2,4-D as far as rapid defoliation?
Well,
And it was like,
it
made
was,
drop, like,
it was 2,4-D and 2,4,5-T.